Good morning, students. We are going into our second set of discussions on bacterial gastrointestinal diseases, and we need to start out with a couple of definitions. First of all, when I say GI tract, it's gastrointestinal tract. Sometimes I go back and forth in between these, and I apologize. It's just habit after being in um, the clinical micro setting, even after so long. Um, and then we also have to talk about an uncomfortable subject for everybody, socially and otherwise, is diarrhea. First of all, I keep forgetting how to spell diarrhea, so I always have to look it up, which is weird. But there are different types of diarrhea. Bloody diarrhea, rice water type diarrhea, and mucousy diarrhea. And a lot of the time, you also have stomach cramping with this. It's not pleasant. And of course, fever and vomiting. All of these symptoms plus other ones are ways that doctors and clinicians will get a clue on what exactly is going on. A lot of the time they miss it because, well, a lot of them didn't care about micro. But in general, this is a set of clues. So when I'm talking about diarrhea and there's clear, a very important effect in the diarrhea, that should be taken of very specific note. Okay, so a lot of these waterborne diseases, foodborne diseases, have a preventable bacterial cause. And this is one of the reasons why I'm really excited about the work of the Gates Foundation, because they're working on very portable, very manageable with low power requirement um, water purification systems for high throughput use for villages and such like that. Because if we can at least give people pure untainted water, that is going to cut out a huge amount of disease transfer in the world and improve the quality of life for everybody, just by taking out some things like cholera, for goodness sakes, or some of the waterborne parasites. This could drastically improve people's lives and decrease the human death everywhere. Um, in general, um, all these are subject to two different types of disease states, either intoxications or infections. Intoxication is basically where there is bacterial toxins that cause um, the actual disease. Usually, when we're talking intoxications, that's pre-made toxin. There are a couple of um, caveats to this, but for the most part, it's just with pre-made toxin. When we're talking about pre-made toxin, at least for bacteria, we're talking about protein toxins um, for the most part. And then with the infections, you have to have the colonization of the bacteria to grow and produce the disease state. So between these two, the food, the amount of illness that you'll get is dependent on how much material you eat, how much of the contaminated food you eat, as well as how long the appearance of symptoms is will affect the incubation period. Okay, so here's what I was talking about before, that how long this affects you will depend on how much of the toxin or the microbe you ingested, as well as the toxin itself. Now, there are ways you can get contaminated food, either improper prepping of that food, either by contamination of the meats while slaughtering, and that's common with um, chicken and beef, or fruits and vegetables are contaminated. They can either have water contamination or somebody fails to wash them and has contamination that was already on them. Um, And then, of course, people being dirty people, they don't wash their hands or not wash it well enough. Again, it is not just baptizing them with some water and the word, and you are going to be clean. That is not in the Bible. What is there is being careful of disease transfer. And in this case, in washing your hands. Hopefully, that is painfully obvious after the last couple um, months. Now, we would be remiss if we did not talk about Staph aureus. Here's our grape-like golden clusters. And remember, the gold refers to the colony morphology, and the grape-like clusters refer to the bacterial morphology. Now, all the symptoms that Staph aureus gives, and they can be a doozy, are through enterotoxins, basically protein toxins that are shed or released and cause the actual food poisoning. You can find Staph aureus in meat and fish products and dairy products, so it's a wide variety. And this is, again, a toxin produced, so it's a protein toxin. And this is usually in a lot of protein, so meats and cheeses and stuff. Um, Luckily enough, this is a short-term illness, so you only have hours instead of days or weeks to worry about whether or not you are close enough to a toilet. Now, the way they usually get infected is through something really gross. There's boils or abscesses from Staph aureus, so not wearing gloves and handling the food, sneezing on it. So sneeze guards and wearing gloves, oddly enough, and washing your hands has helped with the vast majority of infections here. Basically, you refrigerate things, you handle them correctly, and then heat them correctly, and you're fine. Now, one of the ways they do typing of Staph aureus is kind of the reverse of what we were thinking about. Basically, they put bacteria, spread it on a plate, and you've done this, and thankfully not by starting yourselves on fire. You did plate spreading and spread the sample out on the surface of the plate. Okay. Then they put in certain antibodies in kind of a row, drop it on there to see what bacteria it reacts against, or if it reacts against it and binds to that bacteria because it recognizes that type of Staph aureus, it will kill them at that point. So you'll have these clear zones. So this bacteria, the Staph aureus, is reacting to antibody 3 or sera 3, 5, 7, and 14. And between that, you can get this little grid of identification going and say, okay, the antibodies that the strain reacts to are these. Okay, that means it's this type of Staph aureus. And then sometimes you can get information on the correct type of treatment for it, or possibly where they found it, where it's usually contaminating. So it's a very interesting test. They do this in the lab, but a lot of people don't know about it because it's a very specific test and a specifically requested test. And that's usually in um, Staph aureus food outbreaks, like at buffets and stuff like that. They can also do a molecular test now, but we have all the resources for overnight test with that. Okay, Clostridium perfringens you're going to see again later on in gas gangrene, but it is also well known for food poisoning, and this is a false color electron micrograph of Clostridium perfringens. It is starting to have that endospore being developed that with the dipicolimic acid as well as this very sturdy shell around it to protect it against dehydration, radiation, etc. So that is the main way you can get contamination on food is through this spore. Now, that spore means that you have let it sit out long enough so it can make that spore. So if it isn't refrigerated properly, it can start making that spore. If it's sitting out for a long period of time, without being on ice, it can make that spore. So what they'll usually do is rotate their stocks to make sure that the top one isn't always without ice or things like that. But that's why it's found in a lot of meat, poultry, and fish because it's in the environment as it is. Um, and we'll talk about other ways you can get C. perfringens as well. So it needs a lot to overwhelm the gut microflora and grow there. And since it needs time to grow, it's going to take between 8 to 24 hours to appear. So there is, and that one's fairly dangerous. You can get colon segments dying and such like that, but you'll usually have a fever and vomiting and diarrhea then. Now, so Clostridial food poisoning is always Clostridium well, it's always Clostridium perfringens because the other one is a different type of Clostridia food poisoning. Now, Bacillus cereus, you've been working with in the lab. You haven't been licking your fingers or doing anything unsafe, so well done on that. Diarrhea and vomiting, but a lot of the time it's because of contaminated cooked grains. That's right, rice a lot of the time. If you are not having it in, say, a buffet form and cooking it very highly and keeping it at a certain temperature so the bacteria cannot grow, and cannot form spores. Um, that's usually the only way you can do this. If it's heated up, cooked, and then kept at a low temperature or not warm enough to inhibit growth, the spores will actually start growing and start producing the toxin on the grains. This one is fairly violent and very quick. Less than 8 hours, and you're vomiting, and let's just say you're worshiping in the porcelain altar. So, on to one of my favorite topics is Clostridium botulinum. This is an extremely dangerous bacterium, mostly because it produces botulinum neurotoxin, or BONT for short. This is a very, very potent bacterial neurotoxin. It means it focuses on the neurons. It basically stops neurotransmitter release to the muscle. And everybody who is in my Bio 201 class knows this a little bit, but I'm going to go into it a little bit more in detail. Basically, if you get this, you're going to have flaccid paralysis. And then after flaccid paralysis, or part of it, the extreme part is where you cannot breathe. And so you die of asphyxiation. Now, we can put antitoxins, antibodies against the toxin to soak it all up in your bloodstream, but it cannot stop the stuff that is already, the toxin that is already in your neurons from continuing its action. So usually you'll have to be put on life support if it's that bad to let you breathe. Unfortunately, that's usually 1 to 6 months that you're on a ventilator. And a lot of things can happen in 6 months. The most common times I have read documented botulism poisonings lately is either in blood versed ingestion over in Germany, as usual. So a group of them will go down with that because that's not well cooked. And infants below a year and a half, they are fed honey or are right downstream or downwind from a heavy dig site, so it gets well into the air and such, but it's usually honey. Honey has the highest concentration of bacterial or Clostridium spores in nature, um, for whatever reason, and they're one of the few spores that bacteria that can survive that because of the spores. So it is a concentration effect, and the children do not have the normal flora to outcompete the botulism from growing there. That's why adults can eat honey, children can eat honey, but not infants. So wait a little bit, about a year and a half before you give them honey. Waited 3 years with Rhiannon, but I let it go at about 2 with the rest of the kids. Okay, so if you heat the foods for at least 10 minutes, it's going to kill off the Clostridium botulinum. However, the protein is pretty healthy and heat label. Um, and 10 minutes is usually enough, though. Now, we can get botulism through deep tissue wounds. You get these deep tissue wounds sometimes just by getting a softball hit, and it cracks open the skin just enough, even micro cracks, to get a deep tissue infection. That would be an anaerobic environment for Clostridium to grow, but usually infant botulism is the most type. Down here, it shows people and being injected with Botox to look more youthful. It paralyzes those specific nerve endings to the muscles on the face. Other people get it for, well, a vast majority of medical treatments now. So it isn't just for looks, it's a very important treatment. Now, let's see. Where's the floppy? Here. Here's the floppy baby syndrome. You hold a baby like this along the spine, and their head and arms and legs droop back. They usually curl up into a ball or try to poop or something like that. This is the classical flaccid paralysis, and this kid is being intubated and on a respirator because of it. This can be prevented by not eating honey when they're young. We can treat it and clear out the bacteria if it's colonization by penicillin, but they're going to be on a respirator for 1 to 6 months, depending on the subtype of the toxin. The toxin works by getting into the synapse of motor neurons. So it's sending, the neuron is sending signals through fusion of small vesicles charged with acetylcholine and other neurotransmitters. It fuses with the membrane. So here's the vesicle that has the acetylcholine in it. It fuses with the membrane and releases the neurotransmitter into this gap to the muscle, and it binds to a receptor, causing the muscle to spasm or really contract. Now, there are a bunch of proteins on the side, on the cytoplasmic plasma membrane side, as well as on the vesicle, called snare proteins. And these snare proteins are important for fusion. Um, uh, these snare proteins, SNARE, I don't know why it isn't here, are VAMP, SNAP25, and syntaxin. And here's VAMP, and here's syntaxin, and here's SNAP25. And they interact with each other, and they just wait. The vesicle and the plasma membrane just wait here until there's an influx of calcium. And that calcium causes them to, these fusion proteins to have a conformational change, snap backward, and cause fusion with the plasma membrane just a little bit to cause release of acetylcholine. And then they relax, and this releases uh, uh, the now empty vesicle, so it can cycle back and get recharged and filled with acetylcholine, and it goes back through the cycle. What botulism does is cleave these proteins. Oops, there. Sorry. Cleave these proteins, not all the way, but just little bits of it. So there's subtypes A through H right now, and A and E cut SNAP25 at different places. VAMP cuts, or B cuts VAMP. C is said to cut syntax, and that is not true, but it also cuts VAMP. So all these will cause a little bit of cut, and that cut is enough to still allow it to interact, but it will not react to the calcium flux, so it won't cause a conformational change. It's fascinating because these are just alpha helices, just loops, coils of protein, and they react to the calcium in a very specific way. What is interesting is since there is these different types, they recognize different substrates, and they recognize different lengths of it as well as different cut sites. So this was quite an interesting biochemical study for our lab to figure out, and we did figure it out through crazy, crazy means. We tried everything else, and then the brute force approach, and it worked out. Uh, but our postdoc had some heart issues and an ulcer because he worked too hard. But in general, this is how that mechanism works. It's a very fascinating toxin, and I talk about it in a lot of detail in my pathogenesis class. The other thing I should point out is once it's those proteins are cleaved, they can still interact and they just sit here. So even if another vesicle wanted to come in and interact, they can't. So there's this huge gridlock. And the other interesting thing is one enzyme, one molecule of the enzyme of botulism neurotoxin. It's a classical uh, AB toxin. Uh, one one molecule of this will shut down an entire synapse, all the interactions in there for 1 to a number of months. Botulism A is about 6 months, 4 to 6 months. E is 2 to 4. Bot is about 3 to 4, or type B is 3 to 4. So that isn't supposed to happen either. Protein is not supposed to be surviving that long and avoid being broken down and remade. The cells usually break down and remake things every 2 weeks, and we still don't understand why botulism neurotoxin can do this. There's a lot we don't understand about this toxin, how it gets past the intestinal tract into the bloodstream, how it finds its way to the motor neurons, and what is the difference between that and tetanus. This is an important toxin, and here's the receptor, the B subunit, and binding to sialic acid residues. And here's the translocation domain in green, and this is a beautiful representation of the light chain with its zinc uh, atom in the active site. What you can also see is this loop of yellow around, like a belt around the middle of it and going into the active site here. This is how the SNAP25 or VAMP or whatever actually interact with the active site. Usually proteins only interact and recognize the proteins around the active site, but this one requires a huge amount around the entire part of the protein. These are uh, not for Botox, but this is for anthrax. Uh, the botulism neurotoxin research field really it got accelerated through a lot of biodefense research after the anthrax mailing, since anthrax is not great. It has that spore and kills you. But the botulism neurotoxin is exceptionally dangerous if you know how to make it, and not a lot of people do without killing themselves. Okay, I'm going to pause this here and then we'll go on to the final section.